Okay, so welcome to the FOXP1 community webinar. My name is Rob Platica, and I work with the European Organization for Rare Diseases, eurordis, as the online community manager. Today's RareConnect is very pleased to have the first ever FOXP1 community webinar with a team of researchers at the Seaver Autism Center at the Icahn School of Medicine at Mount Sinai in New York, United States. In this webinar, we'll have a chance to hear from these investigators and ask them questions about their work, how it could benefit FOXP1 families, and find out their future plans for FOXP1 research. But first, I wanted to start with some history. We're all here today on this webinar because of an email that Carl sent to me back in December of 2013. I believe right around that time, the Whitneys had just received the genome sequencing results for their son, and we're looking to start a community with other families. So, as you know, we started and created the FOXP1 community, starting just with the Whitney family in 2014, and we've been slowly growing ever since then. If you have any questions during the webinar, you can ask them here in the chat box, and we'll collect them and get to them at the end. Today's agenda will begin with Annie and Carl talking about the FOXP1 community on RareConnect and what we've achieved so far. Next, Joe, Alex, and Paige from the Seaver Autism Center will present characterizing the phenotype in FOXP1. So now over to Carl and Annie.
Carl, this is Annie here. Thank you very much for taking the time to join the webinar today. Welcome especially to Cheryl, Kara, and Kristen. I also want to thank all the folks from the Seaver Center for taking time to speak to our community, and of course to Rob and Marta for helping us set up the community in the first place and help it grow. And I think it's growing very well and quickly. On the screen you can see here a map that shows most of the members. There's actually more members than there are dots, but you can see it's now a global group with concentrations in North America and Europe, but also outside those two areas. So we are very excited to see the membership grow. We now have members in countries, as you can see on the slide, really, starting to span all around the globe. Could you go to the next slide, Rob? So just a couple of small stats about our group. There are 70 members. We have testimonials and stories from a good third of them. We're starting to see a lot of discussions crop up on the group, on the website in the forum topics. And then a large number of visitors, not just members, but other people visiting the website. Perhaps they are family members or people who are newly diagnosed and they're trying to learn more about the FOXP1 condition. But you can see there's quite a lot of web traffic to the site for such a rare condition. And I think this is really one of the great reasons for having the group. It helps bring us together from around the world. Even though we're a small number of people, we've been able to find each other, and I think it's really worth noting and thanking the RareConnect group for their help. So most of the time today will be spent with clinicians and researchers from the Seaver Center talking about the research project that they have been working on. I just wanted to comment that the reason that we invited them to speak today is that we were able to meet with them in April of this year, and we found so they conducted a very broad range of evaluations for our son Jonah. We found the evaluations to be very helpful and insightful, and it's a really good experience, and we were able to learn a lot more about ways that we can help our son, and also we are hoping to help the community by sharing information about Jonah and our family. So I think they will be able to talk more about the research and answer questions, and then I also hope that we can speak a little bit at the end of the time today about a patient registry, which is where we think the next steps lie with this group. The more we can contribute information to a registry, the more we can understand about the range and diversity of developmental experiences for people with FOXP1 changes, mutations, deletions, and additions. So we're really looking forward to the research leading towards a better understanding of the condition, and so we thank everyone in advance for their participation in that project. I think with that, I'll hand it off now to the folks at Seaver, starting with Joe.
Thank you, Carl.
Thank you, Carl.
I'm going to mute my feedback. So I'm going to take you through our work in another rare genetic disorder called Phelan-McDermott syndrome. And the reason is that, we jumped into Phelan-McDermott syndrome in 2010, and I think that through our work and work of other sites, we really kind of changed that field very dramatically in a very short time, as I'll kind of go through. And I think that forms the basis for the rationale for doing something similar in FOXP1. So let me see if I can advance the slides. Yes. So this is a figure that many people now show, which is this idea that if you have a disorder, in this case a disorder of the brain, and you study it, then sooner or later you may understand something about the causes of the disorder. And what's really remarkable is that we're able then, once we have a cause, for example a gene or some change in a protein, then we can create model systems. What that means is we can engineer, genetically modify a mouse in this case, or a rat, or other organism, or cells in culture, so that they have the same kind of mutation that you might find in a patient. And the advantage there, of course, is that it's very easy to study the brain in a model system or a nerve cell in a petri dish, while it's very difficult to study the same thing in a human being. So when you can study the brain in great detail, you can begin to understand what are the changes in the brain that underlie the disorder that you're studying. And that, so far in every case that I could think of, has led to new potential treatments. And it's not to say that they work yet, but it's led to a new insight into possible treatments for these various disorders that have been examined. And presumably, if you can develop some new treatments, you can first of all test them back in your model system. And if they prove to be effective, then you can do clinical trials. And at some point, it comes back to the patients very directly and benefits them in a very direct way. And I'll kind of walk you through this circle for what we've done in Phelan-McDermott syndrome as an example of how powerful this technique is. And it really is unique to academic medicine these days. It's not what pharma is doing, although they're beginning to. It's really it's really become the expertise of academic medical centers. So I mentioned Phelan-McDermott syndrome, which I'll also call PMS. And can you see the mouse, by the way, when I wave it around? Anybody? No. Okay, so I shouldn't wave the mouse around. It's not helping anybody. Oh, now you do. Oh, thank you. Oh, perfect. Well, how do I control it? Oh, yeah. Well.
There you go.
All right, well, here we go. I guess if I click. Okay. So Phelan-McDermott syndrome is a syndrome. It's a genetic condition. It leads to global developmental delay, which is impact on cognitive function. There's also motor abnormalities. There are clear and profound language deficits, and it also presents with autism or autistic traits. So as I said, beginning in 2010, when we first decided to study this disorder and focus on it, we've seen over 40 patients with Phelan-McDermott syndrome and characterized them to identify both strengths and weaknesses. And then ultimately, as I'll show you in a slide, we're able to develop treatment recommendations for the field. So this is an example of a paper that just came out that shows strengths in Phelan-McDermott syndrome. It shows, this is Ting Wang's work here at the Seaver Center, together with Alex Kolevzon and others, including Paige and myself. And basically it shows that unlike what we call idiopathic autism, the individuals with Phelan-McDermott syndrome have a normal response to communicative sounds versus non-communicative sounds. What that means is that all of us, awake or even asleep, parts of our brain that are cued into social sounds and social language activate more when they hear something that sounds like language compared to when something's not language, like a cough, for example. And even though these folks with Phelan-McDermott syndrome have a very hard time with expressive language and some difficulty even with receptive language, their brain function is very normal in terms of how it responds to social sounds. And this is in contrast to autism that we typically see. This is what autism typically looks like, where there's no difference between a non-communicative and a communicative sound. So this is a clear strength in Phelan-McDermott syndrome, and it's an opportunity to take advantage of that strength to enhance communication in the individuals. And we did that by doing brain scans on quite a few individuals with Phelan-McDermott syndrome and comparing the two kinds of sounds. This is the treatment recommendation. It's one of 3 papers that are going to come out from the Seaver Center. Alex was the lead on this, of course, and basically what Carl already said is we have a very kind of extensive workup that we do for genetic disorders, including what you'd expect around psychology and psychiatry, but also genetics, medical and clinical genetics, neurology, endocrinology, kidneys in terms of Phelan-McDermid syndrome, cardiology, gastroenterology, and so on. And so this disorder, which was considered vanishingly rare when we started, didn't really have treatment recommendations, and people really didn't know where to turn. And putting papers like this out first of all, families can access these papers, and they can go to their doctors and say, have we considered looking, for example, at kidney function in our child? And also, the doctors all over the world, the clinicians all over the world, can refer to this when they meet a patient with a specific genetic disorder, and they can know what to look for. Okay, so that comes from seeing many patients at one site. So that's this over here, beginning to study the disorder. And then in terms of discovering the causes, we now know that Phelan-McDermid syndrome is caused by a mutation in a gene called SHANK3. So just like the FOXP1 gene mutation in the FOXP1 associated syndromes, Phelan-McDermid syndrome has a mutation in a different gene called SHANK3. And this was basically the reason we got involved in Phelan-McDermid syndrome is because around 2009 and 2010 it became very clear that there was what the gene was, and so that's how we knew that we could. I'm still waving my mouse around, and I guess how did that green?
If you click the pointer on the top bar, it should appear, and then now you click the green arrow and move it where you want.
Okay. So this is the gene in Phelan-McDermid syndrome, and it's analogous in a sense to the gene FOXP1 in FOXP1 syndromes. So the cause of Phelan-McDermid syndrome, just like the cause of FOXP1 syndrome, but then it goes away. Then the mouse goes away. I don't get it. That's all right. The cause of Phelan-McDermid syndrome is mutation in a specific gene, and so that gives us the power to create model systems. So in the sense, what we were able to do with Phelan-McDermid syndrome is we've made 3 different rodent models. So we basically disrupted the gene in 2 different ways in mice and 1 way in a rat. And between those 3 models, we really can capture a lot of the genetic variation we see in patients. Click the arrow on the second bar, second from the top for the pointer. I'm reading
Right next to
Rob's note. Second from the top. Oh yeah, that one? Oh, good. So we have 3 rodent models that we've created, and as I said, we're able to study the brains of those animals extensively, and if we wanted to, we could study their kidneys or their hearts or anything else. And this is a new technology down here, which is actually taking skin or blood cells from patients and turning them into brain cells, into human brain cells. It's a new technology I'll explain very briefly. But we have 14 pairs of individuals. We have an individual with Phelan-McDermid syndrome and their sibling. So we have 28 of these human brain cell cultures where we can study in a dish, so in cells in a petri dish, we can understand what's going wrong in cells that are actually exactly the same as you have in your brain. And I'll explain that in the next slide. Okay, so this is our first paper on actually mutating the SHANK3 gene in mice. So, Aslan Bozdagi from my lab, and as well as other people, were able to disrupt the gene. And this is a picture of the gene down here. And basically what it shows here is that we've taken out a part of the gene. We've actually genetically modified the mice, so they're missing SHANK3 just like a human patient is. And what we see is, for example, looking over here, that the mice missing 1 copy of the gene, which we call hets, okay, spend less time socially interacting with their peers compared to the wild type or typical mouse. So depending on how many calls they make, these are called ultrasonic vocalizations, so they're calling out to each other. You can see that the animals missing a copy of the gene make fewer calls compared to the typicals, and they spend less time sniffing each other. And that's a social deficit in the mice, just like the social deficits we see in patients. So that gives us a sense that, knocking out this gene, this SHANK3 gene, captures some of the phenotypes we see in patients. And this is, I'll come back to the lower part of I'll come back to these figures in a minute. And this is the other thing I mentioned we're doing. So this is an amazing technology that came out pretty recently, and very quickly the person who developed it got the Nobel Prize. It was the quickest time from developing a new technology to getting the Nobel Prize in history of the Nobel Prizes. Basically, a professor named Yamanaka in Japan showed that if you took cells from a patient, and it could be skin cells or it could be blood cells, you're able to turn them back into what are called induced pluripotent stem cells. Basically, you're able to turn the clock back all the way to the point where they are basically an undifferentiated cell that can turn into any other cell in the body. So a skin cell goes backwards in time, if you will, to being able to that primordial cell that can then be pushed into neuronal progenitor cells, into neurons, which are brain cells, or glial cells, which is another type of brain cell. And then you can study those cells in culture in a petri dish. You can understand how their structure changes. A nerve cell, as you probably know, what it does is signal between cells. So this is the area where it signals to the next cell, and this is the area where it picks up signals from nearby nerve cells, right? And so you're able to actually recreate that morphology in a petri dish. You can understand how the cells communicate with each other, the neuron cells. You can understand how they communicate together in ensembles or circuits, and you can actually throw drugs on this. So if you see, for example, a change in the nerve cell function in the petri dish, either its structure or its connection or its creation of circuits, you can take drugs robotically, actually, and run many thousands and even millions of compounds on those cells in culture to come up with new drugs and therapies. And of course, if those therapies work, then you go back to patient. So this is remarkable because it's the only way to study human brain cells in a dish derived from, it's not derived from the brain, it's derived from blood cells or skin cells. And, for example, if this individual has a FOXP1 mutation, you're basically studying the effect of the FOXP1 mutation that the patient actually had on brain cell function. And when you try a drug on that, you're actually testing the drug on human neurons in culture that are derived effectively from a patient with the disorder. So as I said, in Phelan-McDermid syndrome, we have 14 pairs of these kinds of cells, one from a patient and one from that patient's sibling. So it's just a remarkable new technology that's really a critical part of model systems these days. Okay, so we've got our model systems for Phelan-McDermid syndrome, both the rodents as well as the cells in culture. These are little cells here. And so that gives us the power to understand the changes that are going on in the brain in Phelan-McDermid syndrome. And this is that same slide I showed you earlier, but now looking down here, this tells us that a specific type of receptor that's responsible for communicating between nerve cells in the brain is not working well. This is what it should look like. You put in more and more input and you get stronger and stronger output. But when you're missing SHANK3, that kind of plateaus very rapidly. And in addition, that same receptor is distributed inappropriately. And so this all looks like nerve cells that are immature, that are not fully developed and not fully developed the way they should be. And once you see something like this, we know enough about how the brain works to begin to suggest what therapies might bring this black bar up to where it should be and these black dots up to where they should be. So we begin to understand the mechanisms in the brain that change, that produce the syndrome. So once you begin to understand the biology, then you can say, okay, what might reverse that? What are some of the things that might correct that biological change? And in the mice, back in the mice now, we tested a drug called IGF-1. So this is another paper that came out from the group in 2013. And basically, this is a complicated slide, but it shows, if you this is in blue, is a typical mouse. So this is the connection between nerve cells in the brain. It starts at 100%. You give a special kind of a stimulus, and then it strengthens the connection between those cells, so they're now at 150% of what they were at baseline. If you're missing one copy of the gene, and then that strengthening doesn't persist. It goes away very quickly. It's supposed to last for days, and here it doesn't even last for 2 hours. But if you give those same mice IGF-1, this drug over here, insulin-like growth factor 1, then they look exactly like the typical mice. So we saw a biological change that suggested to us that a certain treatment would be beneficial, and we're able to try that treatment in the mice to see if we were right in our hypothesis. Here's another thing we tested in the mice. This is a test of their motor function. So mice, this is actually on a rotor rod, a spinning rod, and mice can walk on the rod, and as it gets faster and faster they fall off, and this is a question of how long it takes for them to fall. A typical mouse does pretty well, 2 minutes, gets better and better. Missing one copy of the gene, missing one copy of SHANK3, and they do less well, they fall off more rapidly. But if they got the IGF-1 treatment, they do at least as well as the typical mouse. So we're able to say this drug corrects some of the brain function as well as some of the behavior that's associated with our mouse model for Phelan-McDermid syndrome. And so that then gives us the opportunity to try that same drug, and it was chosen because it's already approved for use in kids, to try that same drug in patients. And Alex developed an RCT, a randomized clinical controlled trial of this drug. So the patients either started on placebo or received IGF-1 for 12 weeks. Then there was a 2-week washout period, and if they started on IGF-1, they switched to placebo, and if they started on placebo, they switched to IGF-1. So in the end, everybody in the study got active drug sooner or later. And although this was a really just a pilot study, a first study, the primary thing we focused on, or Alex focused on, and the follow-up, the secondary thing we're focused on, both got better. This is a measure of social withdrawal, and going upwards is an improvement. And what you can see is on drug, folks got a lot better, and on placebo there was no difference. And the same was true for repetitive behaviors. So we've gone from, taking the known gene, introducing that mutation into mice and into stem cells, studying the mice and stem cells, coming up with a potential therapy, testing that therapy in mice, and then coming back ultimately to patients and seeing a difference. This is, as I said, a pilot, so it's just really a first study. Alex and colleagues will finish their second study on the 27th of July, so very soon. So we hope for the very best. And so the idea then is if the second study is equally effective, then we're in a position to, rather than just study the disorder, actually begin to consider seriously treating the disorder. And what I should say, which I don't have slides for, is that once one site, in this case it was actually our site, became an expert on Phelan-McDermid syndrome, we began, of course, to communicate with other groups that either had a few patients or were interested or expected to have patients, and we've created a network, a national network in America with 6 sites. We work very closely with the European sites, Spain, the UK, France, and so on, Germany. And so, all of us now are working together and sharing knowledge, sharing resources for the purposes of moving the field as quickly as possible to the point where we can actually be effective in treatment of Phelan-McDermid syndrome. So, that's the model. This is the model here. And, we believe that that the field, and we can participate in this, the field can do just the same thing for FOXP1. In fact, we have already published a paper on FOXP2. You may know that FOXP1 and FOXP2 work together in their function. So we know a lot about that pathway. We've actually looked at the brain changes when you knock out those partners. And we've started to recruit families to begin to study the disorder. And so this model is what we're proposing for FOXP1, and I will stop there and either take questions now or take questions at the end as they come up. Okay. If not, I'll turn it over to Alex, who will talk about, by the way, we came up, at least for internally, this name, FOXP1-related neurodevelopmental disorders, or FRIEND. But ultimately, different folks will use different names, and the families can kind of weigh in clearly on what they prefer as the title of choice. Alex, want to take it over?
Sure. Thanks, Joe. Thank you. And thanks, Annie and Carl, for inviting us to do this, and for Rob for putting it together. I remember really well, actually, our first webinar for the Phelan-McDermott Syndrome Foundation. And at that time, we were really kind of thinking through what our aims should be in the syndrome in general. What, you know, we had the overall goal, certainly, of trying to drive new treatments forward and improving clinical care. But you know, how we got from Joe's mice to working with the kids was a work in progress. And I think now we have the benefit of 6 years, really, of kind of working through the various issues and having struggled through many different grants and been successful in some of them, and now really collected a lot of data that I think is going to inform the work that we're going to do with FOXP1. And we're really very enthusiastic now, I think, to move into this new space and to draw on our expertise to date. So with that, you know, our overarching aims have always begun with really comprehensively characterizing the clinical picture, what's called the phenotype. And we do that from many different angles, from genetic perspectives. You know, they're similar to Phelan-McDermott syndrome. There's many different kinds of mutations and deletions that can occur with a whole variety of different sizes and chromosomal rearrangements, all of which, you know, result in losing the function of the FOXP1 gene, similar to the SHENK3 gene, as Joe discussed. And that has, you know, a lot of downstream effects on the medical presentation, cognitive presentation, language, behavior. We've always been very interested in how autism manifests. We see FOXP1 just as we see SHENK3 as a autism gene, and so we're interested in, you know, what the prevalence of autism is within the FOXP1 population, and also how does the symptom picture different than what we see in sort of more idiopathic or sort of more classically defined autism. So that'll be our first step also in FOXP1, and I'll go through some of the details there. The other thing that's really critical for us is what's the natural history of this syndrome? How does it change over time? I mean, that's important for a lot of reasons. One, to really enhance our medical monitoring. What are the things that we really want to be looking out for? Is it cardiac problems? Is it seizures? You know, is it language? But also because in preparing for clinical trials, it's very important that we pick the right kinds of outcomes to measure change. And we don't want to be picking an outcome that, you know, may be a moving target in essence. And so looking at the phenotype at multiple different time points is going to be very helpful for us to try to figure out what the natural history is. And then along this line of trying to measure change in the context of treatment, it's really very critical to develop specific measures that are related to the phenotype, that are related to the kinds of changes that we want to see, and not just to rely on more standardized or the existing measures that primarily rely on parent report or clinician observation. We're really very focused in the Seaver Center on developing more objective measures, using things like imaging, as Joe discussed, or as Paige will discuss in a little bit, using things like electrophysiology, and anything that kind of allows us to quantify change in an objective way and doesn't rely on the inherent bias that we see as clinicians or certainly as parents. And that's been a big priority for our center. So that's our kind of main kind of clinical phenotyping aim. And that's all kind of in preparation for clinical trials, which jumps down to 3. And then, of course, within that, we're always collaborating very closely with our basic science colleagues. Joe and the work that he's doing is always extremely valuable in helping us try to figure out what kinds of outcome measures we should be developing, and of course, what kinds of treatments we should be pushing forward. And so his work collecting blood cells, looking at iPSCs, and then looking at the model systems, both behavior of the models and also the way that the brain is sort of operating in terms of electrophysiology. Those things give us a lot of information about what kinds of things we should be doing on the clinical side. So am I able to fast forward this? Oh, Rob, can you fast forward it for me, or tell me how to do it? Yeah, thank you. We started with a review of the literature, and you know, I guess on one hand it's not great that there's not a lot known, but from a literature review perspective, it makes your job a bit easier. So in going through all the literature that used high-resolution genetic techniques to identify deletions in the 3P region that we knew affected FOXP1, I actually only came up with 16 published cases. one of which was published by our colleague Ray Lozano, who's on the call and at the Seaver Center. And Ray has subsequently identified, I think, 2 additional cases that I did not include on this table. But there's not a lot of consistency here, right? And I think the obvious thing to us in reviewing the literature is that this is a very broad and heterogeneous phenotype. So there are a lot of different potential features, and there don't seem to be a lot of very specific ones when it comes to medical complications. Paige will talk a little bit more about behavior, where certainly we see a consistent theme about intellectual disability and language deficits, but I'll leave that to Paige to discuss in more detail. But in terms of what we did find, and I kind of listed in this table, there are some kids with recurring infections. There are a couple of kids where there was obesity, obstructive sleep apnea, low muscle tone, short stature, visual problems, problems in terms of constriction of the gut, feeding difficulties was a relatively common one, hearing problems, I think one child I saw with asthma. Seizures is something that we're really very tuned to, because in a lot of different developmental disorders, seizures can be very prevalent and certainly also affect both cognition and potentially behavior. It doesn't seem to be that prevalent within FOXP1, but it's definitely something that we're going to be keeping an eye on. And then also things like structural brain changes. And similar to Phelan-McDermott syndrome and Schenck in FOXP1, there do seem to be a lot of kids with relatively non-specific structural brain changes, but that's, again, something that we want to monitor, at least to establish a baseline. So things like cysts, for example, if you see those kinds of things grow, it can have an effect on your neurological functioning, and we want to be able to tell what the kind of prevalence of these types of problems are. And then, of course, heart problems are significant, because it's clear that FOXP1, that gene, that protein product, plays a direct role in cardiac muscle. And then kidney problems didn't seem to be a major problem with FOXP1, but it would be still something that we would keep an eye on. So in looking through these kinds of medical problems, it helps us also think about what types of, at the very beginning, really when we know very little, what types of medical assessments we want to be doing at the baseline, and what kinds of problems we want to be monitoring over time. And so I have kind of a very cursory first pass at this that I'll put a slide up on just to sort of show you what direction we're going in. But of course, the work that we're going to do in this syndrome and collecting a lot of data is going to help us refine it in a much more meaningful way. So can you fast forward one for me? Thank you. So dysmorphic features are interesting. This is kind of changes in the way that the face or body are constructed that are not what you would typically see in typically developing folks. And sometimes very specific facial features can be specific to certain syndromes and give us a kind of a clue, even on a very brief clinical examination. That doesn't seem to be the case necessarily in FOXP1, nor is it the case in Phelan-McDermott syndrome. There are many different what's called dysmorphic features associated with the syndrome, but there still don't seem to be anything that we could sort of point to and say, oh, this is a key critical feature. If you see this, you know that it's related potentially to FOXP1. So things that we'll be looking for and things that we'll be examining more closely to perhaps come up with something that is more specific, or at least perhaps a collection of features that are more specific. Things like enlarged head, this kind of frontal hair upsweep. I think I'll show you a couple of pictures. A prominent forehead actually comes up a lot. The openings of the eyes can be short, or they can be turned downward. The space between the eyes can be larger. Sometimes the eyelids can be partially closed. A couple of kids were reported to have sort of a short nose, but with a broad tip, a broad nasal bridge. So these are all these kind of different features that people like Ray Lozano and our clinical genetics folks pay very, very close attention to. And they're things that we see as important in terms of collecting more information. And that'll be a big part of our clinical exam. Okay, next slide, please. And then Paige did a really nice review of all the different behavioral features. And similar to many other types of developmental disorders, we see motor do have disability, although the severity of it is highly variable. And actually, some of the kids that we've seen, and certainly from the literature, many of these kids seem to be relatively high functioning. Autism is certainly a prevalent presentation. A lot of the kids have attentional problems, hyperactivity, impulsivity, those kinds of things. We've seen a few cases, and there are reports in the literature about obsessive-compulsive symptoms or anxiety, aggression seems to be a theme. And here, you can even look at the numbers. The consistency seems to be a little bit higher. Again, these are relatively non-specific, dimensional kind of psychiatric phenotypes, but it does seem that the collection of features seems to be relatively consistent across patients. And this is stuff that we have a lot of experience measuring, and we'll be spending a fair amount of time on in our work with FOXP1. Next slide, please. And so I essentially took what we've developed in Phelan-McDermid syndrome as a parameter, or practice parameter for the medical assessment and the monitoring. We break it down by different fields, because I think one of the challenges that families face is it's almost impossible to kind of get expert care or assessment in one place. They find themselves going to lots of different subspecialties, and often those subspecialties don't communicate with each other very well, and there doesn't seem to be a good kind of algorithm to follow at the time of diagnosis. And that was what we found in Phelan-McDermid syndrome, that parents really were kind of, feeling as if they were out there in the dark, and virtually all the clinicians that they would approach would say, oh, I've never seen a case like this before. And that's not surprising with rare disorders, but we at least wanted to provide something of a roadmap where once the genetic diagnosis is made, what are the kinds of clinical features you want to be looking out for, and what kinds of subsequent assessments should you be doing? And then the next step, of course, is how consistently should you monitor these things? And our natural history studies will inform, what are different periods of development where we could potentially become more vulnerable to developing seizures, for example, or other kinds of problems. And so this is just sort of a first pass. It goes through, what some of the features are in clinical genetics, the types of genetic analyses you want to be doing for sure, including in parents, what kind of psychiatric and psychological workups we tend to do, what kinds of things you want to look out from a neurological perspective, like seizures, of course, as I've mentioned. Endocrinology, like short stature and obesity, seems to come up in a couple of cases. There was at least one case that reported in the literature of hydronephrosis, or sort of a kind of a backup, essentially, of the kidney and bladder. So you want to do a workup in that domain. Congenital heart defects are going to be an important feature that we want to be monitoring closely. Gastroenterological problems, and then a whole host of kind of primary care issues like dental, hearing, and vision. So we see this as really, again, very cursory. This is nothing that we think is worthy of publication at this point, but it's kind of a first step toward one of our broader goals in terms of helping the community develop some practice guidelines. Next slide, please. And so this is an example of what we're going to be doing at the beginning, at least, until we learn more. But all the patients that we see with FOXP1 are going to get physical and neurological exams, clinical genetics evaluations, medical and psychiatric history. We're going to do echocardiograms, which are ultrasound to the heart. Then we're going to do EKGs. We're going to do renal ultrasounds. We're going to do EEGs, which is a little bit more of a in-depth procedure, although a benign one. It's safe. It's just a little bit complicated to do. We do think it's important to try to establish what the prevalence of EEG changes are, and of course, what the prevalence of frank seizures or epilepsy is. We'll do a kind of a routine laboratory blood workup, and height and weight measurement, head circumference, all those kinds of things, as a kind of a core medical battery. I think I have one more slide. I'm going to turn it over to Paige, actually. So thank you very much. I think we will save questions for the end.
Hi, everyone. Thanks for having me. So to kind of pick up where Alex left off, I want to take the opportunity to describe some of the behavioral assessments that we are hoping to collect from families.
Paige, can you hear me? Your volume's a little low.
My volume's low. Is that better?
A little bit. Alex, is that better for you hearing her?
I can put my
A little
headset
bit, yeah.
back. Yeah. Microphone. Is that better?
Yes, I think so. No, now we lost you.
Dave, say something.
No, I heard someone say something, but then I couldn't hear
I
you.
said.
Alex said it. Okay. I thought it was.
I was directing Paige. I don't see Paige's mouth moving.
Sorry.
Oh,
Is that better?
there we go. Yeah.
Okay. All right. Okay. So I want to tell everyone a little bit. Sorry, Joe and I are in the same room. I want to tell everyone a little bit about the behavioral assessments that we'll be collecting, and then I'll share a sample visit schedule so families can have a sense of what they can expect if they are to come to the Seaver Center for an evaluation. So our behavioral assessments have 3 different components of data collection. One involves direct assessment of the child through observations and assessments that look at both behavioral symptoms, cognition, language, neuropsychological functioning. The second component involves standardized assessments with parents that involve parent interviews, so interviews about everything from developmental history, so families will have a lengthy psychiatric evaluation in which they'll be able to describe birth history, developmental history, medical history, psychiatric history, history of symptoms as well as current symptoms in the domains of autism symptoms, ADHD symptoms, sleep, eating, really anything that you could think of that is related to the child's functioning in everyday life. And then we'll also do standardized assessments of adaptive functioning and autism symptoms. And then the third component involves parent report. So these are measures that parents fill out, and it lets us get a kind of a global sense of functioning. So looking at everything from behavior to coordination to a variety of different diagnostic domains that might be relevant to the disorder. So in terms of the different measures that we use, all children who come through get a measure of global cognitive ability, and we have a variety of different measures that we can choose from based on a child's level of functioning to make sure that the measure is most appropriate for that child. So we can do these assessments on individuals who are nonverbal or severely affected, as well as individuals who are speaking fluently and able to respond to task demands. And then when we look at adaptive behavior, that involves a parent interview. So the Vineland is the gold standard interview in that domain, and that measure looks at adaptive functioning, so daily living skills, what a child's actually able to do independently in the areas of personal skills and social skills and coping skills and communication, and looking at kind of how much they can do without the support of others. So not so much what they're capable of doing, but what their day-to-day functioning looks like. And then we're also interested in looking at language. So we know that this is an area that is impaired in FOXP1, particularly expressive language, and the presence of apraxia in many individuals. And so we look at receptive language and expressive language by directly assessing that in the children or individuals who come in with FOXP1. And then we also have parent report measures, and for some of the younger or more severely affected children, we're able to use some direct assessments of language through other measures, such as something called the Mullen, which is a developmental assessment. And then in terms of motor functioning, again, we kind of take this 3-part approach where we look at the child directly, we use standardized parent interviews, and then we also use parent report measures to really get a global sense of motor functioning, and we're interested in looking at both fine motor skills and gross motor skills. And so that's another domain where we're pretty consistently seeing in the literature areas of difficulty. And then the area of autism symptoms is particularly of interest to us, and looking at kind of what traits stand out the most from the children that we've seen so far, and in reviewing the literature, the majority of these individuals do show symptoms of autism but don't necessarily meet full diagnostic criteria. So identifying strengths and weaknesses in terms of social and communication skills as well as repetitive behaviors, and identifying areas that would be meaningful targets of treatment. And so the way that we do that is we do a gold standard assessment. So that involves something called the Autism Diagnostic Observation Schedule, or the ADOS, which is a play-based, semi-structured assessment with the child for younger children. And based on language level, a parent might be in the room for that, so generally something pretty enjoyable for the kids. And then the autism diagnostic interview is a several-hour parent interview looking at developmental history and symptoms that are very specific to autism. And then we also support that with parent report measures. So again, using kind of this three-tiered approach. And then in order to look at a variety of other symptoms, so some of the symptoms that Alex described, such as OCD symptoms, symptoms of aggression or mood issues, anxiety, or anything related to executive functioning, we're also able to assess through parent report measures as well. So the idea is to get a really comprehensive picture of each individual who comes through our center. So going on to the next slide. So something else that the Seaver Center has really taken a prominent kind of stand-in is the area of biomarker discovery. So while parent report measures and interviews and direct observations of patients is really helpful, there's very few objective measures. So measures that aren't impacted by differences in behavior that might happen on a good or a bad day, or kind of reliance on bias measures. And so we're interested in novel measures that are very objective. So looking at measures where we can measure, look at the brain through various means. So one measure is looks at EEGs, and we have several different types of EEG measures that we do here. One looking at the earliest stages of visual processing. They get at some of the mechanisms that we can also look at in the animal models, and then higher order tasks where we can look at areas such as social functioning and attention. And so those are some of the measures that we do here in the clinic. As Joe had mentioned, in Phelan-McDermid syndrome, we've had a lot of success with MRI studies. And MRI studies or imaging studies let us look at both structural abnormalities that might be present in the brain, as well as functional changes that allow us to look at symptoms such as the social domain, for example, as Joe had presented. And then we also do something called diffusion tensor imaging, which looks at white matter tracks in the brain. So again, kind of looking at brain responses from a variety of different measures. And also thinking about eye tracking approaches. So this is another area that allows us to obtain objective data in a very non-invasive way right here in the clinic through different tasks where a child is sitting in front of a computer screen and we're tracking where they're looking at in response to different tasks. So it lets us look at both attention and social engagement and a variety of other areas. And then kind of the last component of our evaluations that fall under these novel potential biomarkers or objective outcome measures is something called the sensory assessment for neurodevelopmental disorders, which we developed here at the Seaver Center. And the SAND is a measure that takes into account both observation and parent interview, and the combination of the two have given us very high, what we call sensitivity and specificity, so a strong ability to identify sensory symptoms in individuals with a variety of neurodevelopmental disorders. And we're specifically interested in looking at where children with different genetic syndromes fall in terms of hyporesponsivity to sensory stimuli versus hyporesponsivity and sensory-seeking behaviors. So with the example of Phelan-McDermid syndrome, for example, we see pretty much global hyporeactivity, so being less responsive to sensory stimuli, versus in individuals with Fragile X syndrome, we see a hypersensitivity. And again, these are areas that give us some information on what might be happening in the brain that can then be studied from a basic science perspective. So to give you a sense of we have many families who travel from long distances, so we try to pack in visits as best as possible, generally 2 to 3 days. we're able to get everything done. So families will come in. They'll have a psychiatric evaluation, so they'll meet with a child and adolescent psychiatrist who will go over that lengthy developmental history that we described. The child generally will then have the ADOS assessment with one of the psychologists. We'll generally take a lunch break from there and then move on to language and motor assessment so that the child's done with testing early afternoon. And then simultaneously, if there's 2 parents available, we'll have a parent also participating in some of the parent interviews. We'll have a dysmorphology and physical exam and a blood draw to do some of the high-level genetic testing that Joe had mentioned, and the IPSCs, collecting IPSCs as well. On the second visit, we'll start the day generally with EEGs and eye tracking and the sensory assessments of some of these kind of novel measures, and then move into cognitive testing with the child while a parent does the ADI, or the Autism Diagnostic Interview. They'll then meet with a neurologist, and then at the end of the day, we'll fill in with some of the medical assessments. So depending on what the child's clinical presentation is, one of the child and adolescent psychiatrists or the neurologist or any of the medical doctors on the team will make recommendations about which specific assessments that individual will be getting. And then all families who come through get a really comprehensive and individualized report based on the findings of the assessment. And so I wanted to take a little bit of time to talk about what types of recommendations we're able to make and how coming to the center can benefit not only research in FOXP1, but also can hopefully be helpful to families from both a clinical and educational perspective. And so each report is really individualized and tailored to the child's needs and to their own presentation. And so we always start with educational recommendations. So we will describe kind of optimal curriculums and ways that curriculums can be modified. So for example, focusing whether a child would benefit from a focus on functional academics or daily living skills. We'll talk about optimal classroom settings, and we'll gather information from you about the child's educational history and whether or not their placements seem appropriate, and so we're able to identify kind of what might be the most optimal classroom for your child. And we'll make recommendations for what targets of treatment should be the focus in speech therapy, whether occupational therapy or physical therapy are needed, and again, using the results from testing to determine what treatment goals are appropriate for your child at a given time. And then we'll also make recommendations about behavioral interventions that might be helpful. So whether it's individual therapy, so modified cognitive behavioral therapy, for example, or simply behavioral therapy to manage symptoms of anxiety or aggression or behavior problems. There's several different behavior management approaches that involve both work in the home, work in the school, or at an outpatient clinic that might be helpful. And then for individuals where social skills deficits are present, we'll make recommendations about social skills training and different approaches that that child might respond best to. And for all of these things, we'll make recommendations also about the frequency of each of these therapies. And then in terms of medical recommendations, the psychiatric evaluation, the neurological evaluation, and the dysmorphology exam will all be written up in the report. And so medical recommendations that span kind of a broad area of disciplines will be made in terms of areas that require follow-up, how often we would recommend meeting with certain practitioners, and anything else related to your child's care. So, for example, recommendations about pharmacology and which types of doctors you should seek out, and any changes in an individual's treatment plan that might be warranted. And I'll let Alex also speak for a minute about that as well, if you have anything to add.
I think you covered it, and I covered it. And I'm happy to answer any questions. Absolutely. Huh? I didn't hear you.
I think it froze for a second.
Okay, sorry, can you repeat what you said, Paige?
So in response to Annie's question, I think we are ready to take questions. On the last slide, we have the contact information for our research coordinator. So if any families are interested in hearing more about participating or scheduling an appointment, Kristen Myron would be the person to reach out to.
Sorry, I forgot to unmute. I'm always doing that. So I see we have a question from Cheryl first. Maybe, Joe, if you want to turn on your webcam, I can read out the questions, and then whether it's for Paige or Alex or Joe or everyone, they can respond. So Cheryl's wondering, for Alex actually, my 16-year-old son is very low functioning. His symptoms have worsened over the years. He has very significant hand and foot neuropathy. He has a C120 duplication with a frame shift effect. And maybe the question is still coming for that. Joe, are you still there? And do you want to turn on your camera too, or? Okay. Great. While Cheryl is finishing her question, please feel free to write your questions there in the chat box as well. And Carl and Annie, if you have something to add too, please feel free. Okay, so Alex, you can probably see Cheryl's questions there, or statements there, but I'll just finish it. Is frame shift rare? You're mentioning many kids are higher functioning.
So first of all,
yeah, go ahead, Jeff.
I don't think we know enough. We don't have enough cases to say what's rare or not. But generally, frame shifts are not that rare. It's a way of the effect of a frame shift is that it disrupts the functioning of the protein that's supposed to be made. And so, you know, you have an extra base, and everything basically shifts in such a way that you don't get a functional protein. So it's a common class of mutations throughout genetic disorders. Yeah.
So, you know, and the question about the relationship between the type of mutation or the number of genes involved in a deletion, for example, and how severe the phenotype is, this is something we've struggled a lot with in Phelan-McDermid syndrome. We certainly have no sense of it all in FOXP1. I think it's an important area because, you know, in the end, if you can help understand which genes in a particular region contribute to the phenotype, or which parts of the phenotype are specific to only FOXP1, it gives you a good sense of you know, perhaps what to monitor. But you know, you need lots and lots and lots of patients and, you know, really well done phenotyping before you can make any good conclusions about the relationship between types of mutations or size of deletions and the severity of the phenotype. So there's another good question about, maybe Rob, you want to read it off? CRISPR. Everybody likes CRISPR, so you can answer that.
Any research attempts to use gene therapy approaches like ISP cells and CRISPR that we're starting to hear more about?
So, you know, the iPSC world is divided into the research domain, where we try and use iPSCs to understand mechanisms and to come up with novel therapies. And then there is another potential use of iPSCs, honestly quite in the distant future, to do gene therapy approaches. And that is being tried. I don't want to say it's not being tried at all in any disorders, but it's being tried in things like the heart, where the cells all kind of look the same, and if you can get some genetically modified material into a heart that kind of replaces damaged tissue, then there's a path forward there. But even that's in its earliest stages. The problem, of course, with the brain is it's much more complicated, and there are many, many different kinds of cells, and every cell is different in the sense that it has to, it tends to have very specific connections and projections and so on. So we are a long way away from gene therapy of genetically kind of putting back in genetically modified cells into the brain. People are trying it, for example, in Parkinson's, where again it's a very localized region of pathology. Specific cells are dying, but certainly not yet in neurodevelopmental disorders. And quite honestly, it's hard to imagine that happening anytime soon. Partly because we really would never know what to inject and where, and a lot of the brain development occurs developmentally, change occurs over time, and you really want to get the corrected function everywhere, not just a place where you're going to put a needle. And so I actually think that the old-fashioned, finding a better drug is actually the better way to go about treatment of the brain, and it's certainly the only way we can realistically do it over the next 10 years. Whether technology changes and things we have real opportunities to do a global gene therapy approach throughout the brain, that may happen in our lifetimes, but we are nowhere near that right now.
So bruising easily, it's not clear to me that that's autoimmune. I think in autism broadly, there does seem to be a higher rate of autoimmune types of diseases that affect the thyroid, for example, in family members. And I'm thinking back on the 16 cases, each of these published papers really just presents 1 to 3 kids, and the extent of the clinical characterization is limited. So I don't recall seeing anything. I think there was at least 1 case of eczema, which is potentially autoimmune, but it certainly wasn't a theme that jumped out. And again, I think as you see more and more cases, more themes may emerge, and that certainly might be one of them. Yeah, that part is clear from like a basic science perspective.
Yeah, another great question, and again, it's some of the same issues with iPSCs. If we knew if there was a very small region of the brain that was missing a protein, and we thought that correcting it would change the phenotype, then that would be a way to go. But again, we don't really know whether, and I suspect we do know, in fact, that FOXP1 mutations affect the brain broadly, not just a specific area. And there are no viruses today that we can kind of get to every cell in the brain. The other side to this too is that it's quite clear with all these what we call gene-sensitive, dosage-sensitive disorders, where too little is a problem, is that too much is often also a problem, although usually a lesser problem. So when with SHANK3, if you have too much, you have a phenotype. With most of the genetic disorders, too much of the same protein or gene is bad too. And so every time we talk about putting something exogenous in, we have to worry about overshooting our mark. And so, that is all of these things, iPSCs as a means of gene therapy, putting in viruses that drive the expression of what's missing. All those are areas of very active research, and they are done quite a bit in model systems. But we're not there yet in terms of anything that might have a positive impact on a global disorder. Again, if there is a specific loss of cells in a certain part of the brain, in Parkinson's as an example, then if we know the protein and we think putting the gene back will make a difference, those studies are ongoing. But in terms of a developmental disorder where we think that the protein needs to be everywhere, that is very much in the research stage and very far away from the clinical trial stage. So Cheryl's asking about age.
So we haven't seen any adults, and I think our typical approach is to take all comers. We're interested in all ages. Once it starts to come down to the point where you do clinical trials, you need to kind of narrow the population. If we do specific studies, it's helpful to narrow the population. But I think as this kind of first pass, we're interested in all ages.
And it also gives us an opportunity to begin to ask whether there's change over time. I mean, Cheryl made the point that the symptoms worsened over the years, and that's important to know. And so getting people at different ages, you can begin to ask questions about changes in symptoms over time. And you can also come back and see the same family a few years later and actually do assessments again. That's what Alex talked about, the natural history.
The adult question is an important one, and we haven't seen it in adults, and I think there's only a couple of adults reported in the literature. That's a very important piece for us, and the follow-up question to that is typically, if you were to intervene in adulthood, let's say we were to find a medicine that we thought would be promising, and we were to intervene in adulthood, but it still have an impact, given how far along they were in their development. And we don't know the answer to that, but the bias is to think that yes, that there still could be an impact, right? There's plenty of research on the basic science side using model systems where you can kind of turn genes on and off in different stages of development and still see significant changes and improvement in the phenotype, including with medicine, when you look at models at different stages of their development. So, we would suspect that the earlier you intervene, the more robust the improvement. But at the same time, we would still be interested in trying to develop treatments that were relevant for adults as well. Absolutely. We see families from all over the world. I saw a family this morning from Dubai. Not with FOXP1, but I'm saying. Yeah.
Thank you, Robin. Thanks, Annie, for putting this together.
Thank you.